Chapter 8, DNA replication and cell division. Now this is a pretty tough chapter. You're going to have to pay close attention. You may have to go online and look up some videos. It'd be a lot easier to do this in person, but I can't this semester, so here we go. Basically, cell division is called mitosis. It has a role in reproduction, growth, and development. The cell division does. There's also meiosis, which is how we make sperm and eggs. But in this chapter, we're going to concentrate on just mitosis and how we make new cells. Basically, the only thing meiosis is used for is for sperm and eggs. Everything else is mitosis. Once a sperm and an egg unite, it becomes a zygote. A zygote is a fertilized egg. It will be a zygote to a certain point, and there's several types of zygotes. It goes through like the morula, the blastula, early blastula, late blastula. For a while, it's going to be called an embryo, and then at a certain stage, it becomes a fetus, and then at birth, it becomes a baby. Now, fetus is not a disparaging word trying to make you feel like it's not worthwhile. A fetus is just a way for scientists to look at developmental biology of any animal species for a certain period to a certain period. All right? It's confusing to try to talk about all these different phases between zygote and the finished product as a baby. All right? You can't do that. Medical science needs to split these phases up. Okay? And that's why we have embryo. That's why we have fetus. That's why we have morula, which is a ball of cells, or blastula, which is a hollow ball of cell. We have these steps, and then the point where it becomes a baby is as soon as it undergoes what is called parturition or birth. Okay? Now, mitosis occurs in every living organism. It can be used to reproduce asexually, basically cloning itself. We see this in single-celled organisms. We see it in bacteria, things like that. It's also used for us to get from here to here. And when you cut yourself, you got to repair that tissue. So mitosis takes care of that. Mitosis even can, in some species, regenerate new limbs. A lot of salamanders, if they lose a leg, can regenerate a new leg. Lizards can regenerate new tails, etc., etc. There's a lot of different ways. Cell death is part of life. As animals go through developmental stages, all right, every vertebrate or chordate, we're vertebrates, undergoes 4 separate stages. At one point, we all have a notochord. At one point, we all have a neural cord. At one point, we all have tails. And at one point, we all have flippers. Okay? But the reason we don't have webbed hands and flippers when we're born as humans is because the cells between the toes undergo cell death. It's called apoptosis. It's where the cell collapses and then it is compacted and disposed of by what is called a macrophage, or a large eater cell. So in the difference between chicks and ducklings, their feet are both webbed at a certain point, but the cells holding the digits together in the chicken die and are devoured by the immune system, and the duckling isn't. All right? For each of the daughter cells from the division, they have to have identical DNA. Every cell has a complete genome in it, and we have to replicate the genome. We have to make a copy. So if we're going to take one set of DNA, we got to make another copy of DNA to go into a nucleus to make 2 cells. And we have several things that are several steps. Okay, so you have to split this double-stranded DNA in order to make copies of it. All right, and we have several different enzymes that play an important role. Helicases unwind and hold the DNA apart. Polymerases create strands. Okay, a polymer means multiple units. So ASE is an enzyme. So these enzymes actually, once we split the strand with the helicase, this is where we match G's up to C's and A's to T's. Ligases will form these on the complementary strand. They'll hook these together, all right, and form this long backbone. And after replication, each molecule will have one parental strand and one daughter strand. Okay. Chargaff's rule. The amount of C will always equal G and the amount of T will always equal A. For example, if C equals 12% of the DNA, then you should know that because G always goes with C, that's cytosine and guanine, guanine will always be 12%. This would amount to 24% of the DNA, which means the other 76% is going to be made up of A and T. So if I give you in a question the amount of G is 20%, how much T, thymine, do you have? That's pretty simple. G is 20%, so C would be 20%, so that's 40% of the total DNA. That leaves 60% left, split between A and T, 30 and 30. Okay, so there you go. When did I get that right? 40? Yeah, 30 and 30. Now, binary fission. Binary fission is a process that basically you just build up one cell and it splits and becomes two identical cells. This is an asexual reproduction. This is a bacillus, a bacterium. Now, in bacteria, they don't have a nucleus. They just have a nucleoid region. Okay. Replicated chromosomes condense before cell division. DNA replication and protein synthesis occur when DNA is loosely packed. Okay. Now, naked DNA, basically, when we want to undergo mitosis, we have to take all this DNA that's spread around and we make these little things called chromosomes, okay, which you can see. And the way we do that, we have these little histones that form little blocks and we wrap the DNA around it to condense our DNA so that we can make copies of our chromosomes. And the chromosomes then split one to either side and forms a new nucleus, and then that's how we get two different cells out of one. The cell cycle works like this. Okay, prophase, metaphase, anaphase, and telophase. That's mitosis. Okay, and then this is basically prophase is where you concentrate all your DNA or your chromosomes. Metaphase, you line them up. This is after you have made copies. Okay, so you have two copies of every chromosome. You line them up, you pull them apart in anaphase, and in telophase you start forming the nuclear membrane again, and then you squeeze the two nuclei in different directions and you get cytokinesis. You will probably want to look up on YouTube a couple videos on mitosis. The G phase, the G1 phase, normal cell function and cell growth. This is just when the cell is being a cell. If it's a bone cell, it's doing what bone cells do. If it's a muscle cell, it's doing what muscle cells do. S phase is when you're making copies of your DNA, and that was this process up here. All right. Then you go to G2 phase. This is additional growth and prepares for division. Then it goes into mitosis again. Every cell is constantly circulating through these different phases. G1, S, and G2 are all part of interphase. There are some exceptions. Certain cells never undergo mitosis. For example, a nerve cell. A nerve cell, instead of after it's formed, it stops all of its copying itself, and it goes into what is called G0 phase. Okay. Now, this is an animal cell. This is a plant cell. There's a couple of little differences, and I have some links that you're going to want to watch on this. Here's the animal cell. So, prophase, you can see the individual pairs of chromosomes. Now, when a chromosome is attached to a copy of itself, it is attached and it is called sister chromatids. Okay? So these sister chromatids will eventually be pulled apart, one to either side, okay, to make two new sets of chromosomes. Now, in metaphase, they all line up in the center. Okay, and you have these little centrioles down here, and these are spindle fibers, and they attach to each side. So this cell has got two pairs of chromosomes. And so 4 chromosomes will go to this end, 4 of these chromosomes will go to that end. All right. So in anaphase, they're pulled apart. Once they're pulled apart, these sister chromatids become chromosomes once again. Then in telophase, they all match up to their correct pair of chromosomes, because as humans, we have 23 pairs of chromosomes for 46 chromosomes, and on those chromosomes, about 20,000 plus gene locations. But in telophase, the nuclear membrane reforms, and so your chromosomes are back in your nuclear membrane, and then the little fibers will squeeze these two cells in two, and they become two daughter cells. Okay? In plants, it works pretty much the same, except for telophase. Instead of these things like a drawstring squeezing these two cells in half, it builds a cell wall between the two things. Here is an actual electron microscope image of a telophase cell, and this little indention here is called the cleavage furrow, and it's splitting it in two. Plant cells have these rigid cell walls. Our animal cells don't have cell walls. We just have plasma membranes. Now, plants also have plasma membranes. But if you will look here and hit open link, it will take you to a little video that will help you to understand how plants and animals undergo mitosis. And then you might want to look for some others. There is some stuff here about stem cells and cloning. I can't really show you those because then the video will get blocked, but these links do play a role. Stem cells, why are stem cells so important? And they're very controversial. Okay, stem cells, depending on what time, there are blood stem cells that can become any kind of blood cell. But trying to program a blood stem cell to become a liver cell doesn't really work very well. In fact, it never really works very well. Okay, and there's a lot of different kinds of stem cells in the body that will become a range of cells, but not any cell. Fetal stem cells do have the ability to become anything, because a fetal stem cell has never differentiated. Okay? It has got the ability, you can take fetal stem cells and put them on nerve tissue that's been damaged, and if you do it soon enough after the damage, they'll reconnect everything perfectly. In here, you will find a video of a little rat that had a damaged spine, and they put fetal stem cells on it, and the rat was able to recover completely. Therapeutic cloning. There are people working, there's a really good video in here of that as well, which you might want to look at. Basically, instead of growing a whole new person, all we grow are organs. So we can take cells, healthy cells. So let's say you have got a disease of the liver. Well, we can find some healthy cells in there. If we can find some healthy ones, we can take them and make a new liver. And it's basically your liver, but it's just not attached. And by therapeutic cloning and making these new body parts, you can take out damaged ones and plug in new ones. And at some point in the future, when you're sick, if you have a problem with your lung or your liver or a bad kidney, they'll take some healthy cells, grow you a new kidney, or lung or whatever, even a heart, and you won't have to deal with all of the immune suppressive drugs because it is your cells, and so your body will accept it as your cells. When you get transplants from other people, the problem is they have different proteins and markers, and like glycoproteins and glycocalyx carbohydrate markers on their cells that will sometimes cause your immune system to attack and kill those cells. And so it's very handy to be able to grow those. And there are people doing it, and in one of these, and there's another video here, in one of these videos, and I'm not sure which is which, it shows an actual lung from an animal that has been cloned from the animal's own tissue. So it has tremendous possibilities. Apoptosis, it's programmed cell death. This is caused by enzymes called caspases, and it gets rid of unneeded cells. Okay, basically you start out as just a few cells, and you become a ball of cells, and then you become a hollow ball of cells, and then you start making things called mesothelium, endothelium, and exothelium. Ectothelium. And those will, different types of tissue in the cell will become different parts of the body. But you're going to go through programming. We all have what is called relic DNA. We basically have got DNA for tails, but it underwent mutations, and so it doesn't produce the right chemicals to maintain your tail throughout your developmental phases until you're born. We have DNA for gills. At one point, every human on this planet had functional gills. It's when you're a fetus. And so, but we get rid of those gill cells because of apoptosis. It takes them out, gets rid of them. Briefly, cancer. Cells, all cancer is when a cell type starts uncontrollably reproducing. Something goes out of whack and you just get cell after cell after cell after cell. And there'll never be one cure for cancer because each type of cancer is somewhat unique. And so you have to come up with treatments. There's lung cancer, and there's bone cancer, and there's skin cancer. And you have to come up with ways to resolve that. And the biggest thing that we're trying to do is figure out what causes it. The best way to treat a disease is to prevent it. And so we find risk factors. And I know what happens. Somebody will say, well, smoking increases your odds of getting cancer at some point in your life. And somebody was, well, I know somebody that lived to be 80-something and they never got lung cancer. Well, each individual has a unique biochemistry, and some people don't. Or they die of something else before the lung cancer really becomes prevalent in their lives. If they live long enough, chances are they will develop some type of lung cancer. Now, there are two types of tumors. There are benign tumors and malignant tumors. Benign tumors are just cancerous growths that are bound by a membrane. Okay, so the little lump, the cyst, gets bigger and bigger and bigger, and then they cut it out. The malignant tumor doesn't have that membrane, and those cells break into the blood cells and the lymph vessels and travel and land in different parts of the body. Okay, and that's a malignant tumor. A benign tumor you can cut out very easily. A malignant tumor is a different story. Here are some of the risk factors that can cause cancers. So if you eat a lot of fruits and vegetables, that reduces your risk of cancer. If you get a lot of vigorous exercise, reduces your chance. If you stay at a proper weight, okay, avoid being obese, increases your chances of not getting cancer. Keep animal fats limited. You got to have some, but we eat more and more animal fats. The amount of animal fats we eat today is like twice as much as we ate in the 70s. Okay? Tobacco. Tobacco, don't start, is your best bet. Never ever use tobacco products. UV radiation. We talked about this in the epithelium chapter, but All a suntan is your body warning you that you are killing cells and increasing your risk of cancer. So it's making more melanin to try to block those mutating UV rays from the sun. So if you really want to look tan, do a spray tan. Okay. Medical exams and self-tests. There's all different kinds to look for lumps or anything like that. Early detection is by far the best. And certain viruses are known to cause cancer, and one of the most important is the papillomavirus, or HPV. It is a sexually transmitted disease, and there is a vaccine for it. It will keep women from getting cervical cancer, this HPV, but you got to take it at like age 10 or 11. And a lot of people who don't understand vaccines and don't understand what it does, it's not birth control. And a lot of people argue again, I don't want my daughter taking this HPV because then she'll just go have sex because she'll be protected. No, that's not what it does. She will be protected against the STD HPV. She can still get pregnant, okay, and you can still talk to her and help shape her moral attitudes towards sex as a parent. But if at some point, even later in life, she has sex with a partner, whether it be a husband, a boyfriend, a fiance, whatever, or a random hookup, and they have HPV, she'll get it and eventually get cervical cancer. So it will protect your children from cancer, which is why I recommend getting the HPV vaccine for young women as kids. Cancer treatments, they try to cut out the cancerous tissue. They can use chemotherapy. They can use radiation. Most of the chemos are not as bad as they were 20 or 30 years ago, but they're still pretty bad. And basically what chemotherapy is, it tries to kill the cancerous tissue. So does radiation. But radiation and chemotherapy can kill you as well, because chemotherapy tends to just be poison. But what they're doing is they're hoping that the poisons will kill the cancerous cells before they kill you. All right? Tumors will mutate, divide very quickly, and develop resistance to drugs. There is a new set, and there's all kinds of new drugs for cancer all the time. And they're testing them and working with them, etc. Here we have traditional chemotherapy, and it slows cancer, generally. But it doesn't eliminate it. But endostatin, this is a group of drugs that target blood vessels, cells, and it basically kills the blood vessel cells, and without blood vessels getting into these tumors, they can't bring important nutrients, so the tumor can't grow. The cells still need resources to undergo mitosis. If they are denied those resources, then they do not have to, then they won't grow. Just that simple. Okay, here's your vocabulary list from chapter 8. Make sure you know it. And that's the end. So again, look at some films, look up some films on this topic, especially DNA replication. I normally do a really big deal on the board, the whiteboards with it, but I don't really have any way of doing that from here. So good luck.